Hello everyone, welcome to today's lecture. In this session, we'll be learning about a widely used protein purification technique, nickel NTA Affinity Chromatography. This method is essential for isolating recombinant proteins, especially those tagged with histidine. Here's what we'll cover today. First, we'll introduce what nickel NTA Affinity Chromatography is and why it's important. Then we'll go over how it works, what's happening at each step from binding to elution. To start off, let's talk about the goal of protein purification. When we produce recombinant proteins in bacteria, we need to isolate the specific protein from all the other cellular contents. That's where affinity chromatography comes in. This method uses a specific interaction between the protein we want and a ligand that's attached to the column resin. This allows us to separate just our target protein from everything else. Nickel NTA chromatography is a type of affinity chromatography that's used for purifying his-tagged proteins. These proteins have a tag of 6 histidine residues added to their N or C terminus. Histidine has a special imidazole ring that strongly binds to metal ions like nickel 2+. So by adding this tag to the protein, we can easily isolate it using a resin that has nickel 2+ ions bound to NTA, which is chelating agent. Now let's look at how the process works step by step. Binding. When we load the sample onto the nickel NTA column, the his-tag proteins bind to the nickel 2+ ions via interaction between the imidazole rings and the metal. Washing. We then use a washing buffer that contains a low concentration of imidazole. This helps wash away non-specific proteins while still keeping the his-tag protein bound to the resin. Elution. Finally, we add a buffer with high concentration of imidazole. This competes with the his tag for nickel 2+ ion binding and causes the target protein to be released from the column. Today, we'll learn how to purify desired protein with nickel NTA chromatography. Thank you for your attention.